First thing I have to do is I just can't give a talk where there's advertising on the board. So I have a real problem with this. So the statement is still true if we write Paxil instead. One of the depressing things about getting old is that you watch people reinventing things that you were angry about in your youth. And I would like the new generation to be really angry about it. But actually, nothing we discussed yesterday and nothing we're going to discuss today is new. It's the same problem with the same issues and the same solutions as we had 10 years ago, as we had 20 years ago, and we had 30 years ago. I know that HIV is a major problem, but it's also true that TB, malaria, diarrhea, measles are major problems. The problems are the same today as they were 10 years ago and 20 years ago. And part of me is wearily depressed about that. It's very hard to pick up the burden and say, this can be changed. And in fact, I'm a strong believer in, I hate to put it in these terms, the creative ignorance of youth. People who have not seen this problem before, who get angry because it really should not be like that. So I'm not going to tell you anything today in the talk I'm going to give about antibiotics, which is any different from anything that you've heard before. The problems have been explained before. The issues have been explained before, and the solutions are the same solutions that have been laid out before society before. Now, because I'm standing up and because unless I make somebody really angry, it's unlikely that anyone's going to interrupt me, I'm going to take this opportunity just to make a couple of reflections on what I heard yesterday before we go on. The first reflection, and I did hear some anger yesterday, and it's about human behavior, where that we should understand. Madam, you expressed your horror of what's happening in Russia. And I agree with you 100%. It's awful. But I would also say that last night I stayed in the ISG. ISG. And the room I stayed in was a very nice room. It had all the basic amenities that I needed to get a night's sleep. However, it was quite clear to me that people had been smoking in that room. And I found that uncomfortable because I don't like cigarette smoke. And let me say that I worked for 13 years in cancer research, and nothing that I did or anybody else did would have anything like the impact of stopping smoking. And people who smoke actually are exactly the same as people who put injections into their arm which potentially can give them AIDS. The only thing which is different is the time scale. You know that smoking, if you are intelligent, put your hands up in this room if you smoke. Do you guys know that there's a correlation between smoking and getting lung cancer? I mean, if you smoke, you've made a decision which is actually very similar to the decision that was made by those kids in Russia. Now, changing people's behavior patterns turns out to be an extraordinarily difficult thing to do. Now, that's just one comment from yesterday. The second comment from yesterday is actually just before I came in the room. Oh, good. So that smoking a cigarette isn't a drug-related activity. And the consequences of smoking that cigarette isn't that you will end up with either a 50% chance of dying of lung cancer or heart-lung disease.
That's not her point. She's saying the reason you smoke a cigarette is not the same as the reason you shot narcotics into your veins. And the fact that both drugs is not the point.
It's also true that in America, if you are caught smoking one cigarette which has got cannabis in it, you are ejected from your job. But if you smoke alcohol, if you drink alcohol, you have a condition which enables you to get help. One is regarded as a disease, the other one is regarded as antisocial behavior. Now, I agree with you that there are complexities. The point I was trying to make was that there are similarities between the two events which lead you to a point where if you start to talk about changing people's behavior as the solution to the problem, it's very difficult to do. So I yield the point that there are different drivers. Nevertheless, I challenge anyone who smokes to explain to me why they do it. The second comment was one that somebody made to me as I was walking in the room. They said, "Oh, I'm glad to see you're back. You made one point last year which I remember." And I said, "What was it?" And he said, "You started off by saying we live in a society where society has chosen to make drugs using the pharmaceutical industry." The point of that was that we use a capitalist solution to making drugs. If we want to change that, I will vote for that, but that is the world in which we live at the moment. So any debate about the issues about making vaccines, about making drugs, has to bear in mind that we are a capitalist society who have chosen that route for making drugs. And if we want to have a debate about capitalism versus socialism, I'm very happy to have that debate. But if we want to have a debate about how we make the system work, which we're currently in, that's a different debate. So now let me talk about antibiotics. And the history of this field is that if you look 20 years ago, people believed that they were on the cusp of solving the problem of infectious disease. One had vaccines, one had antibiotics, and basically the problem was just about to go away. But actually, we all know that this isn't the case, that as we use antibiotics in the population, what you see is the emergence immediately of resistance to the antibiotics that you were using. And this is just a description of data from the USA showing the emergence of resistance to the different classes of antibodies which are used to treat gram-positive infections. And this is just a subset of that data taken for vancomycin resistance in Enterococcus, a serious health problem. And you can see that resistance is reaching very, very large high levels. And there have now been 2 cases in the US of vancomycin-resistant Staph aureus. So it is clear that as you use antibiotics in the way that we use them, you end up with resistance. The cause of this problem, the cause is misuse in part. We still use antibiotics to promote the growth of farm animals. That's a social choice which collectively we have taken, that we will use antibiotics in order to improve the rate of growth of fish, the rate of growth of chickens, and the rate of growth of mammals that we eat. Also, depending on where you are in the world, you can get very easy access, over-the-counter availability of antibiotics. We heard the story yesterday about what happened to Cipro usage in the US. It's the classic case of me versus the rest of society. Because on an individual basis, it may be a better strategy for you not to get vaccinated and rely on the herd immunity of everybody else. And at the antibiotic level, it works in exactly the same way. If you've got a sore throat and you're a doctor, I can tell you that very many doctors pop some antibiotic pills because why should they bother to take the risk of a 10% chance of having a bacterial infection? And that obviously is inappropriate clinical use, where most prescription is against symptomology, it's not against actually what is the cause of the disease, and poor compliance, where partial treatment leads to encouragement of outgrowth of strains with resistance. Those are the social behaviors which cause the problem, but of course we're up against a world where it is equipped to deal with what we throw at it. Basically, bacteria can mutate and they can mate so that they can acquire resistance and transfer resistance. We now know that there is communication between the microbial world so that there has been well-documented transfer of resistance between different groups. And of course, resistance develops very quickly after exposure to the antibiotics. And resistance is just a plane ride away. This just shows the aeroplane routes in the world, and you can see, as many people have said, that we live in a world where disease and the organisms that cause disease can travel very easily. So that's the problem. It's the same problem we've had for a long time. Nothing's changed about the problem, and actually I don't think we've addressed any of the causes whatsoever, even though I could have said this is the cause of antibiotic resistance 20 years ago. What's the solution? The solution's very simple. All you have to do is kill bacteria, and if we had a small molecule which killed all bacteria, was not used a completely different mechanism to the existing antibiotics, and thwarted the development of resistance, then we would be very happy. Unfortunately, it turns out that this is a rather difficult problem. And it also turns out, and let me be very upfront about this, we've failed. Sometimes if you admit the fact that you failed, you can do something about it. When it comes to the production of new classes of antibiotics, we've actually failed. Now, we can have a debate about all the reasons why we've failed, but it would be helpful if we just accepted that we've failed. So if you plot the introduction or discovery of new antibiotics, basically most of what we're working with today comes from the research which was done in the late 60s and the early 70s. And the company I work for, GlaxoSmithKline, is the world's largest seller of prescription antibiotics. And what I've done here is listed the sales. So this is millions of pounds in the year 2001. I've listed the antibiotics on the left. And you can see in red when those antibiotics were first introduced into the market. 81, 87, 83, 88, 72, 69, 85. Where are the new antibiotics? And why haven't we been able to make new antibiotics? Well, there are scientific barriers to that. The scientific barriers, and we should not make light of it. Just as yesterday when we were having a discussion about making a vaccine to HIV, it's not easy. If it was easy, it would be done. So you have to acknowledge the fact that there is a problem. And if you acknowledge the fact there's a problem, then it may be possible to mobilize the appropriate resource to solve the problem which exists. So let me be very upfront about this. Society has failed to make new antibiotics, and in part it is because it is a very difficult problem. Because you want to get a compound which will kill bacteria but will not kill human beings, and bacteria have an enormous amount of genetic variation between them, so the idea of finding molecules which kill all bacteria but don't kill humans is actually technically very difficult. Most of research which had been done looking for new bacteria, antibiotics, was essentially done using the following principle. Let me take as many different extracts or compounds as I possibly can and treat bacteria in one petri dish and see whether we kill the bacteria, and then compare that with treating the same compound and extract on mammalian cells and see whether the mammalian cells are killed. That's the basic underlying principle of how every antibiotic which is on the market today was produced. Wholesale screening of bacteria. And about 10 years ago, GSK and several other companies said, maybe what we've done is we've got it wrong. Maybe what we should do is harness genomic approaches to making antibiotics. If we focus in on targets at the molecular level, it may be possible to break what was clearly a losing streak. And so many people and the science base invested in sequencing bacterial genomes, developing technologies for rapidly testing for essentiality, for rapidly testing for expression during infection of particular targets, and then we try to inhibit those targets. And let me just give you one sort of example of that. In choosing a new target, what would you do? You'd want it to be essential, and you would like that inhibition to abrogate infection. You want the target to be present in as wide a spectrum of bacteria as possible. You don't want it to kill mammals, and one of the things you always have to consider is, actually, is it amenable to small molecule intervention? Here is an example of a target that we've worked on. This is, if you look at bacteria, there are significant differences in the physiology between bacteria and mammalian cells. One of those differences is how bacteria divide. And one of the characteristics is that they use a ring of protein which forms a septum, which is part of the division process. And we wondered whether those proteins could be a reasonable target for intervention. First question was, is FUTZ essential? The answer was yes. Is it present in a wide variety of pathogens? The answer was yes. And does it show selectivity? The answer to those questions was, as I said, yes. It is absolutely essential. It's found in basically all bacteria. But I put in this slide to give you a feel for the molecular problem. This is alignment of sequences of this gene, this essential gene, from a number of different bacteria. And by molecular tricks, we know that this is the essential part of the molecule which is needed for interaction with FUTZ. And if you look at this, as a human molecular geneticist, as I was for many years, I would say this isn't a gene, this is a gene family. And that's exactly what we're dealing with. What I've highlighted here is the bits which are the same, but if you look around the sequence which is conserved, you see that there are many, the residues are not really conserved. Or to put it another way, if you want to inhibit this protein by interacting with a small molecule, you're going to have something which is actually very broad in its specificity. And that brings me back to where are we? 10 years on. 10 years on, what we discovered was that there were many targets which were essential. There were many interesting possibilities, but it didn't actually help us make antibiotics yet. Why not? Well, it turns out that we knew that one of the problems were that bacteria are designed to live in a hostile environment and deal with small molecules that get thrown at them. So we knew that that was a problem. We also knew there was another problem, and that is that very high concentrations of antibiotics are required to kill bacteria, but very high concentrations of foreign organic molecules are toxic to human beings. Those toxicities are seen with the common antibiotics which we use today in terms of rash, liver toxicity. Penicillin causes rash in about 30% of the people who take penicillin. All of the macrolides cause prolongation of QT, which, if that was sufficiently large, would cause heart attacks. Actually, making an antibiotic which you can treat people with at high concentrations, which has no effect on individuals, is difficult. And this just shows you the difference between the sorts of pharmacokinetics, the exposure. Bacteria kill kinetics is both time-dependent and concentration-dependent, whereas the way that exposure occurs in the host you can either have concentration or you can have long half-life, but you can't have both. And if you ask the question, what is it which actually predicts whether an antibiotic will work or not, what predicts is the time above the minimal inhibitory concentration in the animal. So how long you can keep that antibiotic concentration above the MIC. And this goes round to saying that at this moment in time, as far as I know, we actually have made very little progress together. Society, the basic research, has given new targets, but we have been unable so far to convert those into new antibiotics. And just to remind you again, that if we analyze all of the antibiotics which are on the market today, actually the research which led to their discovery was done in the 1960s and 70s. There are also, and this is, again, nothing new, social barriers to the production of antibiotics. We decided to use companies to make antibiotics. That was a social decision. In some countries, my country, for example, we chose to use a social approach for hospitals. We said the hospitals and the roads will be done centrally and owned by all of society. Actually, last year we decided we'd change that rule, and now some of the hospitals are going to be owned privately. It's a social decision that has to be taken. So the production of antibiotics is a commercial activity, and therefore it obeys commercial rules. If you ask the question then, well, what are the commercial drivers? It turns out that as the single largest pharmaceutical market, antibacterials is pretty good. It looks like it would be attractive. 8% of the global pharmaceutical revenues come from antibacterials. However, if you look at the top 20 pharmaceutical products, i.e. the top selling drugs in the world, only one of those, Augmentin, is in the top 20 so far in the year 2002, and by the end of 2002 it will not be in the top 20 because it has gone off patent in the US, and so this will disappear off this list. So there will not be a single antibiotic in the top 20 best-selling drugs. And if you look at the market growth for antibiotics, what you find is, and this is value now, not prescription rates, what you find is that the average pharmaceutical growth rate is 10%, but the average growth rate for antibacterials is 3.8%. What that means is that people looking to say, where should we invest, are going to choose to invest in things other than antibacterials. Remember, this is market-driven production of things that society say they want. And if you look at the volumes for antibiotics, in one level, we're winning the battle because the use of antibiotics is decreasing in the world. So having been very pessimistic about changing behavior, actually we're finally beginning to see a drop-off in the volumes. This isn't the value now, the volumes. But again, if you're looking at a marketplace and the number of units which are being sold is decreasing, then you have an incentive not to work in that particular area. And to follow that pathway, let me just explain the different parts of the antibiotic market. This is based on global sales. And the one thing I draw your attention to is most of the value is in antibiotics which are given to people in the community. This is the antibiotic you get when you have a chest infection, a sore throat, you go and see your doctor. And this value is mostly driven by the fact that nobody actually checks to see whether you need the antibiotic or not. That's the reality of the situation. The biggest problem from a social point of view is down in the highly resistant gram-positives. That's £600 million worldwide. That, in pharmaceutical terms, is a marginal market. There are several antibiotics in this class, and if you took that whole market, you would only have 600 million. Compare that with the worldwide sales of Paxil. The worldwide sales of Paxil last year were 1.5 billion pounds. So about a third of what you would get with this antidepressant. That's market forces. And it's not cheap. So you have to make decisions about the investment that you're going to make. We can have an argument about what it really costs to develop a new antibiotic. This doesn't include the social contribution in the science base. This is just talking about what it costs a company to produce. There are all sorts of different ways of calculating the number, but it's going to be somewhere between a third and a billion, a third of a billion and $1 billion, something of where in there. And it takes you 10 years to 20 years to produce a new drug, so it costs you a lot of money, and that investment is for a long time. This is something which is extraordinarily expensive, and this is why society decided that it's better to use the capitalist approach rather than use the social approach of saying we will take that cost centrally. There have been a couple of antibiotics which have reached the market in the last few years, and I just thought I would put them up to balance the picture. There has been a new macrolide, a new subclass of macrolide, which was is in the process of getting worldwide regulatory acceptance. This actually is no new science. It's tweaking an old class of antibiotics. when this new antibiotic was taken to the FDA, they were worried about liver toxicity and QT prolongation. And they said to Aventis, the only way you can convince us that you can sell this drug is if you do a trial on 24,000 patients. So if you bring a new antibiotic along and hope to get regulatory acceptance, you're going to have to spend a large amount of money. Why do they do that? They say, perfectly reasonably, if it's a community antibiotic, it's going to be given to a lot of people who don't need it. Therefore, the risk-benefit ratio has to be towards safety, not towards efficacy. Remember, you go to the doctor, you've got a sore throat, the doctor says, have you got a temperature? You say yes, you get the antibiotic. The other new antibiotic, and this is a new class of marketed antibiotic, Zyvox. This treats vancomycin resistance in Enterococcus. This is not a new antibiotic. The basic research, this class of antibiotics has been known for 20 years, but this is the first marketed example of this antibiotic. This goes into that 600 million marketplace. What did the FDA do? The FDA said, we'll let you sell it, but actually we're very concerned because this, from a social position, is a very new tool. And we're going to try and restrict the use of this antibiotic because it's so valuable to society to have this new tool. So essentially, they have placed on top of Zyvox a mandated surveillance program, a program looking for the emergence of resistance. Why? Because you would want to control the prescription of this drug if it turns out it looks like you're going to lose it very quickly. The commercial consequences of that is, why would you bother? Because you're not going to make any money with a new drug which treats this class. Please remember I said this is a social decision. We can choose to do something differently, but this is what's happening at the moment. So what are the solutions? I think the first thing is, and this is a statement of fact, if you look at the major pharmaceutical companies, they are basically not doing anything like the intensity of research and development in antibacterials that they did 5 years ago, 10 years ago, and 20 years ago. The problem has turned out to be very difficult, and the value in the market is somewhere else. There is a solution to that, and it turns out the solution is that biotech companies always love antibacterial research and oncology research because it's dead simple in the early stages, because all you have to do is take a petri dish of bacteria and see whether your compound kills them or not. So I think that we will continue to explore target-driven antibacterial research in the biotechnology sector. Whether that will lead to translation into new drugs, we will have to see. So far, it hasn't. And this is just a way of saying what I said before. The nice thing about antibacterial research and oncology is it's very cheap up front until it becomes expensive later on. So it's made for biotechnology companies. The second possibility, which I've emphasized all the way through, is we could change the rules. We could find a way of altering the value proposition such that it would encourage people to do more of this research if we were prepared to pay the cost. Now, how you wish to change the rules is a social decision. It is possible to change the economic rules for companies, as has been done for orphan diseases, where you give more value to companies by giving them more protection if they do research for orphan diseases. Or we could take a decision to actually do central, as was hinted at yesterday, we could take the decision to have the National Institute for making, or the World Institute for making new antibiotics. So I'd just like to finish by thanking a number of my colleagues. I have to confess that I faked all of this today. I do have a degree in microbiology. However, it's many years since I pushed bacteria around on a Petri dish. So I had a lot of help in bringing the slides together from Alan Olive, David Payne, Lynn Marks, David Coccecchio. So the slides are theirs, but the mistakes are mine. Thank you very much.
Thank you very much, Peter, for this provocative presentation. I think we'll have questions for Peter. All right.
First, a clarification question and then a question. The clarification is the numbers of revenue and sales, did any of that include veterinary use, or is that all entirely human?
No, it's all human.
So if you look at the equation and add in the profit incentive associated with veterinary use, does the balance shift at all in favor of production of antimicrobials?
No, this is just a rule of the world that we're actually much more interested in human beings than we are in animal welfare, and therefore the value is in human use.
My understanding is by far the bulk of the marketplace is growth promoter use of antibiotics.
I know. Are we talking about volume or value?
No, I'm talking about how much money the drug company makes off of it, therefore an incentive to go into it.
As I said, are we talking about volume or value? Value means how much money you make. Volume is how much you produce. There's no value in doing the volume business for animals. So that is it's a different business altogether. Margins are very smaller. The research component is very small, and there is no drive to make antibiotics which are for agricultural use. So the economics are quite different.
Okay, then my question is, 2 weeks ago at the Whitehead Institute Symposium, Tony Fauci, is there anybody here who doesn't know who Tony Fauci is? Tony Fauci vowed...
He's better known in America than in Europe, but never mind.
But he commands about $4 billion budget for basic R&D in infectious diseases. And he has control... He has control of $1.75 billion bioterrorism research budget. At the Whitehead, he said, by the year 2012, we will have a universal antimicrobial. What do you think of that?
Obviously, he knows things that I don't know, and I'm greatly reassured.
Bertolt Rutz from the European Patent Office. You commented on the changing the social values. Do you think there's any sense in prolonging the patent duration for giving an incentive to develop new antibiotics?
That would be one of the ways that you could change the balance. I have to be very careful to comment in a room with journalists for, person from drug industry says you shouldn't increase patent life. I probably get shot when I went back home. Speaking as purely a consumer, I'm not sure that that's the approach that I would take to solving this problem. But it would be a potential pathway. So one could say, if you actually produced a new class of antibiotic against a target which had never yielded an antibiotic before, we will actually reward you for having done that in a way which is over and above the current system. So that would be a way of doing it.
Could I just comment directly on that? There have been some discussions over the years at WHO with the IFPMA, the Pharmaceutical Manufacturers Association, not looking at antibacterials or antibiotics, but looking at the more neglected diseases where 600 million would be a huge global market if it existed. I think the summary conclusions were that where you're essentially dealing with a small market, patent extensions, they can be useful when you're at the borderline between a valid market and an invalid market. but of themselves, they're not going to probably lead to a huge increase in incentive for companies to invest in expensive R&D. So I think at the interface of market sizes, there may be value. If you're talking about a huge quantum leap up, maybe less so.
One comment which I would make, and again, it's not a new problem, that let's say a new antibiotic class was discovered, which actually was, the magic molecule we're talking about there. Would you give it to people in Africa? I mean, that's the same problem again, but there will be a lot of social debate about what is the appropriate use of this reagent and how it should be used. And I pick the words very carefully when I would say it might be better for a company not to get involved in that debate even, because you can't win it. So because you can't win that discussion, maybe you shouldn't get involved.
You were talking about the side effects of the old and potential new antibiotics. Do you think that the antibiotics that hit the market decades ago would win FDA approval nowadays? And is this also a restriction of getting new drugs on the market?
And the answer is yes, of course, it's a restriction on getting new drugs on the market, but that quite reasonably, the FDA says, prove to me that the antibiotic that you now want to give to millions of people is as safe as the antibiotic which we've been giving to millions of people for the last 20 years. That will not change until people die when they go in hospital, because when you go in hospital, you're more likely to die from the infection than if you didn't go to hospital. The driver isn't there yet to change the risk-benefit ratio, which is why Aventis had to test 24,000 people for a new antibiotic. Yeah.
Hi, Peter. I'm sorry you're not still an angry young man. And I had an uncle who had to flee from the States to Canada during McCarthyite times, and he used to say, if you're not a Marxist at 20, something's wrong. If you're still a Marxist at 40, something's wrong. So we all get that way. And my question comes back to the one just addressed in a sense about niche markets. And I mean, contrary to Tony Fauci, contrary to the big farmers, I mean, one could say, well, if we look at antivirals, which are supposed to be in their infancy, we don't pretend that we must have a drug that knocks herpes viruses and retroviruses and hepatitis C. And we say, well, these are different and they have different molecular mechanisms. Let's fit the drug to the bug. So why isn't there a niche market for a mere 600 million?
There is, and I said, but you have to realize the choices that people make when they say, should I go after a market which is several billion for my product, or 600 million if I've got this money to invest? Because essentially the investment is the same for either market. And so that's where the choice comes about how you spend that money. The other part is that an antibiotic which only treats one species of bacteria, I think we can do. You have to know that the person is infected with that species of bacteria. And actually, that's not how we treat patients. Most antibiotics are given to people who don't have bacterial diseases.
We have 4 more questions already, and I think that's about the time that Peter's going to leave, and I try to go by order as people were indicating that.
A very related question. You have shown us that one of the bottlenecks in the new molecular targeted antibiotics is that, of course, the sites are not conserved between families, right? And you've shown us that this is possibly the reason why, until now, all the genomics explosion has yet failed, because you have not been able to bring new drugs to the market. So do you think that that would change if you then would start developing targeted therapies against single species of bacteria? And
from our practical results, we have produced reagents which show narrow spectrum activities against bacteria for which there isn't a market for a narrow spectrum agent. So the answer is yes, it makes it easier. Just like it's much easier to make a drug which works in humans but doesn't work in all mammals or all vertebrates. And it's probably even wider diversity when you're talking about bacteria than comparing us with plants.
So it does work, but it's a problem of the market, basically.
It's a combination.
Thank you. My name is Peter Schoch from Basel. You have nicely and at length depicted the problem. You were very brief in indicating a solution. There was just one sentence. The value proposition should be changed. Could you expand on that a little bit more?
I think that we could get more, we could increase the chances of success if we put more resources and more focus into the problem. But if you put more resources into the problem, then people would want to see a larger return. So somewhere society has to pay in order to get that to occur. That could occur either by changing the rules under which companies work, and we talked about that a little bit, or we could take a step back and say, as a world society, we think this is so important that we are collectively going to solve this problem. And I'm not sure that the world has that type of drive to actually do it, but it's possible. Maybe, as Robin says, angry young men and women could change the world.
One more quick question up here.
Again, the question about changing the rules is a question if we still err too much on the side of safety. I know of at least one case where a completely new antibiotic compound has been found, has been developed into a product to treat chronic open wounds in diabetic patients. Yeah. It has an extremely broad spectrum, works completely different to any other existing companies. The small biotech company that developed the thing went partnered with Johnson & Johnson, I think, successfully completed all clinical trials, and the FDA rejected it on the ground that it did not work better than the existing one. It worked only as good as.
We had a product there as well, and I have to tell you that nothing actually works. So you get in a rather complicated situation when you try and do those trials, because they want an active comparator, but actually none of the comparators work. But yes, the answer is things will change for sure when there is sufficient drive that this rises up the list, so people are more concerned about this than cardiovascular disease, diabetes, cancer, and it becomes the #1 issue in society. And people are prepared then to pay more money and take more risk in order to get access to something which may cure them of what otherwise would be a lethal condition. So, in a sense, I think you could say that the driver is already there. The bugs eventually will do it for us. When people start dying, your kids start dying when they break their leg because going into hospital is a lethal event, this will change the rules.
Hi, Aratu Castro. I'm not angry now. And it's interesting to note that the volume of drugs for cattle is not that important in value terms when we remember that actually Merck's ivermectin donation program for river blindness is possible because Merck's returns for producing ivermectin for animals actually are high enough so that they can donate it for poor people. And
I need for balance, I have to say that GSK donates albendazole which we don't have an animal business, but we also donate that free for filarial disease. So there are a number of examples.
But yeah, but in ivermectin's case, it is evident that the donation is possible because of the market that it generates with cattle. And then when we see that, and we also see that TB drugs are, I think they're still considered in the orphan list in the United States, when we know that it's the second major killer. And the third thing that I wanted to remember is that we looked at, PAHO conducted a study of the cost of antiretrovirals in Latin America. Yeah. And many of the antiretrovirals have the highest cost in Haiti, which is the poorest country in Latin America. And those three examples, the ivermectin, the TB drugs, the cost of antiretrovirals in Latin America, remind me that there is a problem with the definition of market. Yeah, of and you know, if we believe that increased need or increased demand should lower costs, and we look at the cost of antiretrovirals in Haiti, we think, you know, there's a market failure, or we're just defining market in a very wrong way.
Yeah, everything you said is correct. But let me give you an HIV example. I work for a company which is called GlaxoSmithKline. The reason it's called GlaxoSmithKline was because it came from two companies. Each of those companies came from two companies and so on, but I don't need to go there. On one arm, there was a company called SmithKline Beecham, and that company was the first company to clone and express the protease from HIV. And we had peptide mimetics in SmithKline Beecham, which actually worked against HIV. And the decision was made, please don't write this down if there's any journalists here. The decision was made not to go forward with that because the cost of the drug, which would be made, would mean that there would be no profit for the company, and they would be under intense social pressure to donate that drug. So I'm not saying that was the right decision, that was the social pressure. On the other hand, the other company which bears the heritage I work for decided they did exactly the same thing, they did the same experiment, and they decided to go forward with drugs which, not the protease inhibitors, but to go forward with drugs which would work against HIV. And what happened? They got into very large debates about whether they should give the drug away or they shouldn't give the drug away in Africa. Now, I actually think that the activity of angry people has moved the equation such that the major pharmaceutical companies are now much more responsive than they were before. They've realized that that's an essential part of the cost of doing business, and I welcome that. But I still worry at the back of my mind that you can focus on heart treatment for HIV and completely miss the point of the curve which was shown by Robin yesterday. And now I'm going to go watch Arsenal beat Newcastle